Two.
Okay, good afternoon, everyone, and welcome to our grade 12 spring school exam preparation classes. And today we will be doing the final session. So we're going to look at now for the next 2 hours, we're going to cover Life Science Paper 2. And these classes are presented by the Cape Town Science Centre in collaboration with Astra Energy. So, guys, before we get started, there are some ground rules. So, you guys know your mics and your cameras are both turned off. That is to not allow any disturbances for anyone in this lesson. As well, guys, if you do have any questions, please pop them in the chat, and then myself or my co host will get to that as soon as possible. Then, very important, guys, I need you guys to write the following in the chat for me, please. Please state your name, your school's name, as well as the number of people viewing from your device. So, can you guys just please do that throughout the duration of the session? Okay. So let's get started. So, obviously, guys, before you start answering your questions, first and foremost, you need to read through the questions or the instructions nice and clearly and thoroughly. Make sure you understand what is being asked of you and so forth. Okay, so guys, what's going to happen now is we're going to jump straight into understanding experimental variables. So, the hypothesis. So, what is a hypothesis? It's basically a possible solution to a problem or an assumption that is proposed. So, hypotheses are testable through experiments or observations that you make, and hypothesis always falsifiable, meaning it can be proven wrong or it can be proven right. Okay. So with a hypothesis, there are relationships between two things. The relationships occur between the independent and the dependent variable. The independent variable is what we change. We, as the scientists, we change that, and that causes an effect on the dependent variable. So it responds to the change. Okay, then throughout your experiment, you always have a control variable, constant variable that remains unchanged. So with your scientific method, there are two things that I like to look at. So this is the validity and the reliability of an experiment. So let's look at the first one. We have validity. Validity asks how accurate are your results. It basically asks how accurately a method measures what is supposed to be measured. So to ensure the validity of your results, you need to specify what is being measured, and you need to ensure that your methods and instruments that you use are accurate and correct. So, for example, if you need to measure maybe like water or something, you're obviously going to use perhaps some sort of measuring device. Obviously, it won't be a ruler, for example. So, next up, guys, I'm going to look at reliability. So reliability looks at how consistent are your results, basically how consistently a method measures something. So also reliability wants to ensure that someone can redo your experiment. So to ensure reliability, you need to follow the exact same procedure every time you do the experiment. You can't change your experiment or the process of experiment as you redo it. That's going to obviously make your results unreliable. And also another way to ensure the reliability of an experiment is to constantly repeat the experiment to see if you get the same results. Another way is to increase your sample size. So instead of using one person for an experiment, you instead need to use a much larger sample size, maybe 10 to 50 people. Then it's also very important to repeat your experiment, because when you repeat your experiment, you can then calculate an average for the experiment. Okay. So that was just covering basic tips and ideas for your scientific method question that you would get in your exams. But with that type of questions, it takes a lot of practice to be able to identify what is the independent variable and what is the dependent variable. As well as for the validity and reliability part of that question, you would then more just look at what they have given you in the information. In there, it would state anything to do with validity or reliability of the experiment. Okay, so we're going to jump straight into the first main topic of paper 2, and that is DNA code of life. Okay, and that accounts for about 27 marks in your exam. So basically, guys, I'm just going to cover some of the main topics under DNA. I'm not really going to focus on anything that has to do with the makeup of DNA or the structure or anything to do with the DNA nucleotide. I'm assuming you guys should know that. That's very basic information. So instead, we're just going to look at DNA replication. So what is DNA replication? It is basically, sorry guys, it's basically the duplication of the DNA molecule to form two identical copies. So replication occurs during the interphase of the cell cycle and occurs only in the nucleus of the cell. So this occurs to both plant and animal cells. So functions of DNA. So usually they would ask functions of DNA, they would ask this question for about maybe 2 marks in the exam. So first you can say that DNA makes up genes which carry hereditary information. And then you can also say that DNA contains coded information for protein synthesis, okay? So why is DNA important? So during mitotic cell division, one mother cell divides into 2 daughter cells, right? So at the end, each daughter cell has the identical DNA composition to the mother cell, okay? So that question, the important DNA replication, that's also maybe about 2 marks in your exam as well. Okay, so now let's look at the process of DNA replication, and this would generally count for about 6 marks in your exam. So when you are mentioning DNA replication, you need to mention it in chronological order. Don't confuse the different parts. Please mention in chronological order. Okay, so first part of DNA replication, you have the DNA double helix over here that unwinds. Then next step, you have your weak hydrogen bonds that occur between the nitrogenous bases. They break and your 2 DNA strands begin to separate. So now once the 2 DNA strands have separated, they now will both serve as templates. So what happens? Free nucleotides, so your adenine, thymine, guanine, cytosine, they then occur in the nucleoplasm, and they are now used to build a DNA strand onto each of the 2 original DNA strands. So obviously the complementary nitrogenous bases, they start attaching to each other. So adenine binds to thymine and guanine binds to cytosine. Then at the end of DNA replication, what you have is 2 identical DNA molecules. Each molecule consists of 1 original strand and 1 new strand. Okay, so that is basically one of the most important, or the few of the most important points when it comes to DNA replication. Okay, guys, so let's move on to protein synthesis. So again, I'm not really going to go through the RNA nucleotide. That I'm assuming you guys should know is very basic things over there. So now let's move on to protein synthesis. So protein synthesis, what happens here? It's the process whereby proteins are manufactured in living cells. So next up, where does protein synthesis occur? So it occurs in the nucleus. Only during transcription occurs in the nucleus. And then it also occurs at the ribosomes in the cytoplasm during translation. So nucleic acids, DNA, and RNA control the synthesis of the proteins. Okay, so they also like to ask the question, where does protein synthesis occur? So just take note of that. And then functions of RNA, there's just one. It plays a role in protein synthesis. Okay, so protein synthesis, we know, occurs in 2 main steps. First one, transcription of DNA. So the synthesis of mRNA from a DNA template. So remember, transcription occurs in the nucleus. Then we have translation of RNA to proteins. So this is the process of converting the information carried by the mRNA to the correct sequence of amino acids to form a particular protein. So in the exam, they don't usually ask you to mention both processes. Instead, they will ask you a couple of questions before this, and then they will ask you explain transcription or explain translation, and each of these questions would go for about 6 marks each. So they don't really ask you both processes, only one process would usually be asked. Okay, so let's look at these two processes separately, and let's look at it look through it step by step. So protein synthesis, we have transcription. So remember, transcription occurs in the nucleus. So again, it's a process. So remember, you need to mention this process in chronological order, in a correct order. Don't mix things up because that won't allow you the marks. Okay, so first thing, again, it starts off similar to DNA replication. So what you have is that the double helix DNA begins to unwind and it splits over here. Then you have your weak hydrogen bonds break to form two separate strands. Then one DNA strand will now act as a template for forming your mRNA, which is this over here. And this will be formed using the free RNA nucleotides from the nucleoplasm. So obviously we know DNA and RNA differ in the fact that RNA is single-stranded, DNA is double-stranded, and RNA, instead of having thymine, RNA has the nitrogenous base of uracil. Okay, then, so once your single mRNA strand is formed, the mRNA now has the coded message for protein synthesis, and the nitrogenous bases on the mRNA are known as codons. So 3 nitrogenous bases make up your codons. So the mRNA will then now leave the nucleus through the nuclear pore located over here into the cytoplasm where it attaches to the ribosome. So the ribosome is this blue-green thing here at the back. Okay, so that is the process of transcription. Okay, so now we're going to move on to translation. So there you can see the mRNA moves into the cytoplasm. So let's look at translation. So translation occurs in the cytoplasm, and its main point where it occurs is on the ribosome. So what happens first is you have your single mRNA molecule that was formed that attaches to the ribosome. Then the mRNA, like we said, provides the code for the linking of amino acids to form a specific protein. Okay, then over here, this is your tRNA molecule. Now, each tRNA carries a specific amino acid. So these 3 lines over here, those are the nitrogenous bases of the tRNA, and we refer to them as the anticodon. The 3 nitrogen bases on the tRNA make up the anticodon. So when the anticodon on the tRNA matches the codon on the mRNA, then tRNA brings the required amino acid to the ribosome. Okay. So at the end, the amino acids become attached to each other by peptide bonds to form the required protein. Okay, perfect. So now we covered a few important parts of the DNA code of life. So now we're going to move on to a question from paper 2, June 2022. Okay, so sorry about that, guys. Okay, so the question reads, the diagram below represents the process of protein synthesis in a cell. Then the table below shows some tRNA anticodons with their corresponding amino acids. Okay. Okay, so just erase all of that, guys. Apologies for that. Okay. So apologies, guys. The answer's already there. Okay. Right. So what does it say over here? It says, name the DNA base triplet that codes for serine. So we only have the tRNA, the anticodon. So the anticodon for serine is UCG. So how did we get that? The answer over here we have is TCG. So all you simply have to do is you basically are just changing, because obviously over here we have the DNA. Sorry guys, just to change this thing over here. Okay, so we're going to look at setting. So you have your anticodon, which is UCG, and that is tRNA, right? So they're asking us to change it to the DNA base triplet. So we need to change it to DNA. So obviously the only difference, or one of the differences we can see now here between the RNA or the tRNA and DNA is the uracil. So uracil is the difference between RNA and DNA. So all you simply do is you just change uracil to that over there to thymine, and there you go. And that's how you get your DNA base triplet that codes for serine. So basically all you're doing is you're replacing the RNA base with its DNA counterpart. Okay. Then next up, next question, it asks the first two amino acids coded for by molecule Z in the diagram. The molecule is read from left to right. So let's have a look over here. Let's just highlight it to make it easier for you guys to see. So first two amino acids coded for by molecule Z. So molecule Z is our mRNA, because as it's shown in the diagram, that is your mRNA that is attached to the ribosome. So they're asking us to look at that one over there, UAC, first amino acids. And the second one they're asking us to look at is GUA. So let's just change that to another color. GUA. So those are the two molecules that we are now going to look at. Okay, so we're looking at molecules here, that is the mRNA, right? So we are now looking at the codon. So now we need to change that into the anticodon, right? So what's going to happen is we're going to change this. So the complement to U is A, the complement to A is U, and the complement to C is G. So that is going to be tyrosine as we have over here. Then the second amino acid that we're going to look at is GUA. GUA. And the complement to G is going to be C. The complement to U is going to be A. And the complement to A is going to be U. So CAU is going to be valine. So that is all simply how you do that question over there, guys. Then, next question. What is the change in the sequence of nitrogenous bases in a DNA molecule called? That is just simply called a gene mutation. Okay. And that only counts for 1 mark, so they just want the name. They don't want any definitions there. So always keep track of the mark allocation for each question. Okay. Then, last question in the section. They say over here, the codon CUU, the last codon. So let's just quickly highlight that again. So they say that the last codon, CUU, which is that over there on molecule Z, changed to CCU. Explain the effect it would have on this particular protein molecule. So let's quickly look at this a bit more in detail. So the last codon, CUU. So that is codon, meaning it's mRNA. So if we were to convert that CUU to an anticodon, the anticodon would be GAA. And if you look at GAA, that would code for the amino acid leucine. Okay. Then, so now they said a change from GAA to CCU. So now that's the codon CCU. If we change that to the anticodon, what would the anticodon be? Complement of C is G, and it would be G again. Complement of U is A. So GGA would be proline. So what we note from this is that there is now the change in the protein that occurred. So let's just have a look and see how we would write this out. So it says the codon will not be GGA, as we mentioned over there. So the codon will be GGA, will be GGA and not GAA. That means the last amino acid would be proline instead of leucine, as we have shown over here, resulting either a different amino acid, a different protein forming, or no protein forming at all. Okay, so that is the topic of DNA that we have now covered, guys. And next up, we're going to move on to meiosis, and I count to 21 marks. So DNA and meiosis are the smaller parts of your paper 2. It's a bit more of the study part of it. But yeah, we're going to move on to the other bulkier sections in a little bit. Okay, so meiosis. So let's just break down meiosis into its different parts, and I'm just going to highlight the important parts of meiosis again. So meiosis. So what is meiosis? It's a type of cell division where a diploid cell, a somatic cell, undergoes 2 cell divisions and divides to form at the end 4 haploid cells, gamete or your sex cells, right? So remember, meiosis undergoes 2 cell divisions, meiosis 1 and meiosis 2. So where does meiosis take place? It takes place in the testes and ovaries of animals as well as humans, and in the anthers and ovules of plants. So they also would ask this question, where does meiosis occur, for about 1 or 2 marks in the exam. Okay, so a nice way to remember meiosis is by this mnemonic imp mat over here. So let's look at what the first one says. So we have the interphase, I for in between, the part of the life cycle of the cell that is in between cell divisions. So next up we have prophase, P standing for preparation, where the chromosomes become prepare for meiosis by untangling and becoming clearly visible. So crossing over takes place in prophase, but remember it only takes place in prophase 1. Then metaphase, M for middle. So that one's quite easy to remember. The chromosomes move to the middle all along the equator, right? And that occurs obviously in both meiosis 1 and meiosis 2. Then anaphase, A for apart, the chromosomes or chromosomes in the case of meiosis 1 and in meiosis 2, chromatids move apart or move to opposite poles. Then telophase, T for terminal, the final phase of meiosis 1 and meiosis 2. Okay, so now guys, we're just going to look at the next one. That's going to be the differences between meiosis 1 and meiosis 2. So they also like to ask these types of questions. So let's quickly just look at it. So if they're asking you to like tabulate the differences between meiosis 1 and meiosis 2, for example, you can say crossing over takes place here in meiosis 1 and crossing over does not take place in meiosis 2. When you are labeling it like this, please don't say crossing over takes place here, but then in the meiosis 1 you say that, but then by meiosis 2 you're saying 4 cells are formed. It needs to correspond with each other. 2 columns or rows or lines need to correspond with what they are saying to each other. Okay, so let's look at the first one. So it says the chromosomes arrange at the equator of the cell in homologous pairs. So that is now referring to metaphase 1 in meiosis 1. So this is the chromosomes arranged at the equator of the cell in homologous pairs, as you can see over there. Then in meiosis 2, what you have is chromosomes line up at the equator of the cell individually. So they're no longer in homologous pairs, they are individually aligned along the equator. Then over here it says whole chromosomes move to opposite poles of the cell. That is now referring to anaphase 1, because remember, anaphase A for moving apart. So whole chromosomes move to opposite poles of the cell. And then in anaphase 2, now we have daughter chromosomes or chromatids. They are now moving apart again to opposite poles of the cell. Then, at the end of meiosis 1, you have 2 cells formed at the end of this division, as indicated in telophase 1 over here. And then at the end of telophase 2, you have 4 cells at the end of this division. Now in meiosis 2, you have the chromosome number remains the same during meiosis 2, but in meiosis 1, the chromosome number is halved. Then In meiosis 1, crossing over takes place in prophase 1 over here, but in meiosis 2, no crossing over takes place. Okay, so moving on to the next one. So now we're going to look at the importance of meiosis. So why is meiosis important? Basically, it produces haploid gametes. Then, meiosis is also important because the halving effect of meiosis overcomes the doubling effect of fertilization. So basically, in meiosis, you move from diploid to a haploid number. So what you can say is that meiosis ensures that each gamete, each sperm or egg cell contains half the number of chromosomes, so meaning they're haploid, compared to the parent cell, which is diploid. So when fertilization basically occurs, the fusion of the haploid gametes, egg and sperm cell, restores the diploid number in the zygote. So this balance basically between meiosis and fertilization is crucial for maintaining a consistent number of chromosomes across multiple generations. Okay, so importance of meiosis, why it's also important. It is important for genetic variation. So they also like to ask this question. They would ask, how does meiosis influence or cause genetic variation? And you need to be able to answer this question pretty well and just write the following. So, as we know, crossing over takes place, and crossing over generally occurs in prophase 1. So, what happens over there is you have your homologous pair, and then what happens is the two basically, the inner chromatids of each homologous chromosome pair, they touch each other, and the point at which they touch each other is either called the chiasma for singular, or the chiasmata, which is plural. So at the point where these two inner chromatids, where they cross over, that is basically where the chromatid segments, they actually break off. And basically they are now exchanging genetic material. Then you end up with gametes at the end that are now genetically different from, chromosomes now that are genetically different from each other. Okay, then another way in which genetic variation can be determined or brought about is through random assortment of chromosomes at the equator. Now, this obviously occurs, if things are at the equator, obviously occurs at metaphase 1. So what happens during this process? So we have two diagrams over here. So random assortment during metaphase 1. So what happens? Diagram A and diagram B. So they both show different arrangements of the homologous chromosomes at the equator. So these homologous chromosomes in both diagrams are arranged very differently. So now in diagrams, in the diagram next to it, it says gene combination of gametes produced according to arrangement A and arrangement B. So as you guys will note, that the gametes in both arrangement A and B, they all have different gene combinations. And this shows you now that this is how genetic variation can be brought about. And now these genetic or gene combinations are obviously brought about by the homologous pairs that are arranged differently along the equator. Another way in which genetic variation can also be brought about is through random fertilization, where it's the possibility of which sperm will fuse with which egg cell during fertilization. So those are the three ways in which genetic variation can be brought about, either through crossing over, random fertilization, or random assortment of chromosomes on the equator. Okay, so those are some of the important points that I like to question you guys on. So it's either going to be, well, in this case, importance of meiosis. They'll most likely ask you, explain how meiosis brings about genetic variation or variation. So you guys can name all of these things over here. Okay, next up, abnormal meiosis. So this is also another question they like to ask, usually for either between 4 to 6 marks. So abnormal meiosis or non-disjunction. So this happens in anaphase 1, where there may be non-disjunction of homologous chromosomes into separate chromosomes. So non-disjunction, that refers to the failure of chromosome pairs to separate during meiosis. So this can also happen in anaphase 2, where there may be non-disjunction of chromosomes into single-stranded chromosomes. So let's have a look at the diagram here on the side. So we obviously have normal meiosis taking place on the one side, and then we have abnormal meiosis taking place, but this is now occurring in anaphase 1, where we have homologous chromosomes that did not separate. So the diagram shows you when non-disjunction occurs during the first meiotic division. So here we have one cell ends up with both pairs of chromosomes, while the other only receives one. This results in an abnormal distribution of chromosomes. Then we have another abnormal meiosis taking place, where this one occurs in meiosis 2 in anaphase 2. So on this one, it shows non-disjunction happening in meiosis 2, and one chromosome now fails to separate properly, which then results in one cell with an extra chromosome and another cell missing a chromosome. Okay. So if there is non-disjunction of chromosome pair 21, that's usually where abnormal meiosis takes place, it leads to the formation of an abnormal gamete with an extra copy of chromosome 21. So now, if there is fusion between a normal gamete with 23 chromosomes and an abnormal gamete with an extra copy of chromosome 21, this will then lead to Down syndrome. So at the end, the zygote will have 47 chromosomes instead of 46. A person with Down syndrome will have 3 chromosomes at chromosome pair 21 in every somatic cell. So this is how you guys would write out the process of abnormal meiosis if that is asked you guys in the exam. At the bottom of this diagram here, this is just listing the gametes at the end of meiosis. You don't have to know how the chromosomes are distributed into the individual gametes. You just more need to focus on the process of abnormal meiosis, how it occurs in anaphase 1, anaphase 2, and what is the result of that is that someone will, a zygote will end up having Down syndrome when it is, when the normal gamete is fused with the abnormal gamete. So that's all you guys need to know in terms of abnormal meiosis. Okay, then moving on to mitosis and meiosis. So now we're going to just look at the similarities and the differences between them. So starting with the similarities between mitosis and meiosis, both processes are cell division processes. Both require DNA replication to occur during interphase before the division phases begin. And obviously they both have the following phases: prophase, metaphase, anaphase, and telophase. So that's quite simple and easy to remember. But now let's look at the differences. So there are quite a few differences between mitosis and meiosis. So mitosis occurs in body cells, somatic cells, and meiosis occurs in the reproductive organs. So at the end of meiosis, the chromosome number is halved, and in mitosis, the chromosome number remains constant. Mitosis, where you have 2 cells that are formed, and these cells that are genetically identical to one another and to the parent cell. However, in meiosis, 4 cells are formed, as we saw now, and the 4 cells at the end are genetically different from each other and from the parent. So they're genetically different because crossing over took place in meiosis in prophase 1, which caused or allowed for genetic variation. But in mitosis, no crossing over took place, meaning there's no genetic variation, because the cells are identical to one another and to the parent. So mitosis has only 1 cell division. Meiosis has 2 cell divisions, meiosis 1 and meiosis 2. Then in meiosis, homologous chromosomes arrange in pairs during prophase 1. And in mitosis, no pairing of homologous chromosomes take place. Okay, so guys, that is the main points in meiosis that you need to take note of. Yeah, okay. So those were now the less bulky topics of paper 2, but now we're going to move on to the bit more bulkier topics, and that is going to be genetics and inheritance. And that accounts for double the marks of the previous two sections, and that is about 48 marks. Okay, so let's get started. I'm hoping you guys went through some of the questions that I sent you guys, but let's get into it. So we're going to look at now paper 2, November 2020. So the question reads, a homozygous purple flowering plant is crossed with a pink flowering plant to produce the F1 generation. One of the F1 plants is crossed with a pink flowering plant to produce the F2 generation. Which one of the following is the correct phenotypic ratio of the F2 generation? Okay, so with this question, guys, I'm going to show you guys how to work it out to get to the correct answer. Okay, so let's just highlight the important points of it, because when I do these types of questions, I like to highlight and know exactly what I'm working with. So we have a homozygous purple flowering plant, and that is crossed with a pink flowering plant to produce the F1 generation. Then we have the F1 plant is crossed with a pink flowering plant to produce the F2 generation. So which one of the following is the correct phenotypic ratio of the F2 generation? Okay, so let's write this out. So we have a homozygous purple plant. That is the phenotype, and that's going to be crossed with a pink plant. Okay, cool. So they say that the purple plant is homozygous, so we know capital P, and that's going to be crossed with a pink plant. And we can deduce from the question that pink is the recessive allele and purple is the dominant allele. So capital P is dominant, I'm just going to do that. And then small p is the recessive allele. Okay, so if we cross these two plants in the F1 generation, what we are going to have is a heterozygous purple plant. Okay, so now they also said in the second part of the question, the F1 plant is going to be crossed with a pink plant to produce the F2 generation. So if we take the heterozygous F1 plant and we cross it with a pink plant, we're going to get the F2 generation. So now, just to show you guys the cross and to explain how we're going to get the answer, I'm going to draw a Punnett square for you guys, just like that. Okay, so I prefer doing Punnett squares instead of doing the crosses because it's a bit more neater and you can see exactly what is happening. So in your Punnett square, you need your gametes. So we're gonna, it doesn't matter where you put your gametes on your Punnett square. Okay, as long as the gametes are one parent on one side and the gametes on the other parent on the other side. So if we're gonna cross these two over here, starting with this one and this one, it's gonna be a capital P and a small p. We cross this one again, the small one over here. We're gonna get that, and then we're gonna cross the next set of gametes between the two small p's over here. I'm gonna cross in this one and this one with the two small p's over there. So now, what is the phenotypes that we're looking at? At the end, we're gonna have these two plants over here are heterozygous, purple. So we're gonna have two purple plants. And then we have these two plants over here. They are homozygous recessive, meaning they are pink plants. And we also have then two pink plants. Okay, so we have a ratio of 2:2. Now if we look at the answers provided, there is no ratio of two purple plants to two pink plants. But now this, remember, is a ratio. So what we're going to do is we're going to simplify, because the ratio you always simplify as far as possible. So you simplify this, you just basically divide by 2 on each side. Then you're going to have one purple plant and one pink plant. And now that looks similar to the answer A. So for 1.1.6, your answer is A, one purple and one pink plant. Okay. Then moving on to the next question. So the question reads, inheritance by multiple alleles in genetics refers to A, two alleles that influence two characteristics, B, more than two alleles that influence one characteristic, C, one allele that influences more than one characteristic, and D, more than two alleles that influence two characteristics. So this is simply just definitions that you guys need to know. And now the answer for 1.1.6 is going to be B, just simply definitions that you guys need to know. So inheritance by multiple alleles in genetics refers to more than two alleles that influence one characteristic. And you would know this answer if you have looked at blood groups. So blood groups are an example of a gene that has multiple alleles. So the alleles occur at the same locus on a particular homologous chromosome pair. Because you know with blood groups, basically it has three alleles, or the allele A, allele B, and the recessive allele O. Okay. So now we're going to look at a few questions on blood groups. Okay. So it reads, a man with blood group AB and a woman who is heterozygous for blood group B plan to have children. How many alleles control the inheritance of blood groups? And again, this is just from studying knowledge. The answer over there is 3, because the allele that you're going to look at is going to be A, B, and the recessive allele I. Okay. Then the next question asks, describe the type of dominance that occurs in the inheritance of blood group B in the woman. So let's just highlight again. Okay, so a man has blood group AB and a woman is heterozygous for blood group B. Okay. So the type of dominance occurs in the inheritance of blood group B in the woman. So the woman's heterozygous for blood group B, meaning her genotype can only be this over here. So we know that blood group B, or allele B, is a dominant allele. And this blood group over here, blood group O, is a recessive allele. So blood group B is completely dominant over blood group O. If the woman, for example, in the man's case, his genotype is blood group AB, and his blood group, there's no allele that is completely dominant over the other. In this case, allele A and allele B are co-dominant. Okay. So in the case of the woman, is heterozygous for blood group B. Blood group B is completely dominant over blood group O, or allele O, and allele O is a recessive allele. So how would you write that out? It's complete dominance. The allele for blood group B is completely dominant or dominant over the allele for blood group O, which is recessive. Okay. Right. So let's move on to the next question. So a man with blood group AB and a woman who is heterozygous for blood group B, and they basically plan to have children. So now they're saying use a genetic class to show all the possible genotypes and phenotypes of the children. Okay. So how would we write this out? So I'm going to write it out exactly how you would in your exam. Then I'm going to explain to you guys where exactly you would find your marks. Okay. So let's start off first. So what you're going to say first is you're going to say P1. And you're going to list the phenotypes So the phenotypes of the man is going to be blood group. I'm just going to say blood group AB. And the phenotype of the woman is blood group B. Then what is the genotype? We know the genotype for the man can only be one genotype. It's going to be that over there. And then the genotype for the woman, because she's heterozygous for blood group B, it's going to be the allele B with the recessive allele O. So now we are going to cross these two pairs together. So like I said, I prefer doing the Punnett square. So let's just draw that out. Okay. Here we go. So obviously what we're going to write on the side is meiosis. Meiosis occurred to form the different gametes that we're going to mention. So you can have your gametes. So the gametes of the male is IA, allele A and allele B. The gametes of the female is allele B and the recessive allele I. So we're going to cross the allele A with allele B. So one of the gametes, one of the phenotypes is going to be blood group AB. Then we're going to cross allele A with the recessive allele I. And then one of the phenotypes over there would be blood group A. Then we're going to cross allele B with this allele B over here. Then you're going to have blood group B as well. Then we're going to cross blood group B over there with blood group I or blood group O. And then you're going to have completely dominant blood group B. So now, obviously, meiosis occurred, and then you have your gametes, which you listed this over here and over here. So now, when you cross your different gametes, fertilization took place. So you would write that over there. So now, most importantly, you have your F1 generation. So what is the phenotype that was determined? So allele A and allele gamete A and gamete B crossed. So you got blood group AB, gamete A and gamete I crossed. So you got blood group A because blood group A is completely dominant over blood group O. And then you had another cross, you ended up with blood group B over here, homozygous for blood group B, and then over here you have homozygous again for blood group B. Then, okay, I don't have any more space, so I'm going to write it here on the side. Then what you'd also write is, sorry, not F1, P1, and F1, and meiosis and fertilization. So just by writing these two lines, you get 2 marks. Okay, so I'm just going to indicate where you guys will get your marks. So over here, by mentioning the phenotype, that is 1 mark. By mentioning the genotype is another mark. By mentioning your gametes, that is another mark. By mentioning the phenotype of the F1 generation, that's another mark. By mentioning P1 and F1, another mark, and meiosis and fertilization, that's another mark. So in total, it's 1, 2, 3, 4, 5, 6 marks. And is how you get your 6 marks for your genetic cross. Okay, guys, we're just gonna take a quick break so I can have a sip of water, and then we're gonna continue with the rest of the genetics section of this session. Okay, guys, so let's get back to it. Right. So continuing with genetics. So we're looking at now paper 2, November 2019. Okay. So what we're looking at is a pedigree diagram over here. So it says the diagram below shows the pattern of inheritance of a disorder. So obviously, when you're starting with these types of questions, you need to have a look at the key first to tell you what you are looking at. So the square unshaded represents male that is not affected. The circle unshaded represents female not affected, but the circle shaded in represents female affected. So the question asks, one can conclude that the disorder is caused by, and they list all the options. So what we're going to do is we're going to kind of go through the options and we're going to eliminate the ones that are not possible. Okay. So let's get started. So if we are going to look at question A, it says, so first things first, I just want to kind of create a key. So I'm going to say my key is going to be D, standing for dominant allele, and then small d standing for or representing recessive allele. So they're saying in A, so the disorder is caused by the recessive allele with both parents heterozygous. So both parents heterozygous, D crossed with D, and the disorder is caused on the recessive allele, small d, meaning that means that both parents would not be affected, right? Because they're heterozygous. And if you do end up crossing this, you would have one of the F1 generations to have been affected. Okay, so I can just draw that cross for you guys if anyone's uncertain about this. I know this is only a multiple choice question and a 1-mark question, but I think it's sometimes always best just to draw things out to make you fully understand what is happening. So if we cross the two parents, and let's have a look what type of genotype the child would have. So if we're going to cross the two capital T's, the dominant allele, that would be 1. Crossing this allele, that gamete to that gamete, it's going to be that and that over there. And cross this one with that one, it's going to be that over there. So all of these 3 in the F1 generation would be unaffected, right? But now, if we cross this one over here with that one over there, this is the only one in the F1 generation that would be affected. Therefore, A could possibly be an answer. So let's just make a mark over there. Okay. So let's take that all away. Okay. So now let's look at what B says. So B says it is always caused by a dominant allele with both parents heterozygous. So if that was the case, both parents would have been affected. We don't have to at all look at the F1 generation. Therefore, B can't be the answer because if you look at the picture they've given us, neither of the parents are affected in the situation. So the answer can't be B. Okay, let's look at C. So this order is caused by the recessive allele with one parent homozygous recessive, meaning this is the one parent's genotype, while the other parent is heterozygous. Again, if we look at C, it's caused on the recessive allele, meaning one parent had to have been affected. If we look at the diagram, neither parent is affected, therefore the answer can't be C. Could the answer be D, dominant allele? So they say one parent is heterozygous and the other parent is homozygous recessive, meaning this order is caused by the dominant allele, which is D over there. So what that's telling us and what the heterozygous parent had to have been affected. But again, in the diagram, if we look, neither parent is affected. Therefore, the answer can't be D. So we end up with the answer being A. Okay, hope you guys understand that. So that's only a 1-mark question, but I thought it'd be best to explain it to you guys in that setting. Okay. Okay, so let's look at a bit more of a longer type of pedigree diagram. Okay. So it says one type of deafness in humans is carried on a single allele. Diagram below shows the inheritance of deafness in a family. So obviously the unshaded individuals with circles and squares, that is the hearing individuals, whereas the shaded circles and squares, that is the deaf individuals. So now before we move on to the questions, let's just look and analyze the diagram, because I like to have an idea of what I'm working with before I move on to the questions. So how many generations do we see here? So we see one over here. This next set over here is the second generation. This third set over here, third generation. Okay. Then just by looking at the diagram, we don't have any information on genotypes of the individuals, but just by looking at the diagram, we can actually see what is the dominant characteristic amongst the individuals, because what you can clearly see, there are more hearing individuals and deaf individuals. Okay. So let's actually move on to the questions and see what they ask us. So how many generations are in this pedigree diagram? And as you could see in the previous question, there are 3, as you've noted. So they are. Okay, just change this color quickly over here. So there are 3 children of Paul and Lizzie that are able to hear. So let's quickly look at the diagram over here. So how many children of Paul and Lizzie are able to hear? So Paul and Lizzie is over there. We can see that they have 1 hearing male, or sorry, 2 hearing males, and they have 1 female child, and that female child is the only deaf child. So meaning children of Paul and Lizzie are able to hear is 2. There. Okay, let's move on to the next question. So which type is dominant? As we looked at and we're analyzing the diagram, we could see, that there are more unshaded blocks than shaded blocks, meaning that hearing is the dominant trait and deaf is the recessive trait. So we can just say there, hearing is the dominant trait. Okay. Then next question, they say using the offspring of Bob and Anne to explain your answer to question 2.4.2. So when they say explain your answer, you always need to explain it in a give a statement and give a reason. That's the way in which you can say when you state something, you need to give a reason as to why you're saying what you say. Okay. So let's just look at Bob and Anne and see what's happening by them. Okay. So Bob and Anne are both hearing individuals. They crossed and they ended up with 2 hearing children and 1 deaf child. Okay. So what we can do is, since let's actually just create genotypes for these people to make it just a bit more simpler to understand what is going on. Sorry guys, having a bit of technical things happening on the side. Okay. Sorry guys, okay, back good. So let's create a bit of a genotype for the individuals. So let's say for hearing individuals, we're going to use A as the dominant allele. And for the deaf individuals, we're going to say small a is going to be the recessive allele. Okay, so because one of the children of Bob and Anne is deaf, we can definitely say that their genotype has to be homozygous recessive. Okay. And then what that also tells us, obviously, because one of their children is homozygous recessive, that means this child had to have inherited either of these 2 alleles, actually both of these alleles, from their parents. Meaning the genotype of Bob and Anne, each of them had to be, they each had to have the one issue, both of them have to have a recessive allele. But remember, we're saying that the disorder is carried on the recessive allele, but neither parents actually have the disorder. Therefore, we can say because they don't have the disorder, they should also have a dominant allele. So what we could actually say is that both parents are heterozygous for the disorder. Okay. So let's see how we would actually write that out a bit more clearly. So we can say both, obviously you can clearly say both Bob and Anne can hear. So like I said, the child who is deaf has to have the genotype AA because we said AA represents the recessive allele. And therefore, if that child had the recessive allele, it means that they need to have inherited the recessive allele from either parent, meaning each parent carries an allele for deafness. And since the dominant allele A, that basically masks the expression of the recessive allele small a for deafness. Therefore, both parents need to have been heterozygous for the condition. Okay. So let's move on to the next part of that question. So it says, use the letter A to represent the dominant allele and the letter small a for the recessive allele to give all the possible genotypes for a hearing individual. So if an individual was hearing, that would be the first possible genotype. And the second possible genotype would be 2 small a's. Okay, next question. It says use a genetic cross to show how gender in human offspring is determined by 6 chromosomes of the parents. Okay. So again, this is just a very simple genetic cross that you need to do. So I'll write this one out again. So this is also again accounting for 6 marks. So you have P1, that is your phenotype that you're going to list. Then you're going to say, so they're saying a genetic cross show gender in human offspring. So obviously you're going to cross a male and you're going to cross a female. Then the genotype of the male is XY and the genotype of the female is XX. Just like that. Then again, we're just going to draw the Punnett square. And then you're going to list your gametes. So we're going to put the male at the top and the female on the side. And then you're just going to write over the gametes. So obviously, in order to form the gametes, meiosis took place. And then the gametes are going to cross, which will result in fertilization. Okay, so the gametes are going to cross. You're going to end up with XX, XX, and then end up with XY, and you're going to end up again with XY. So the phenotype would be 50% males and 50% females. So you can say 50% males and 50% females. Okay, so that is the result of your F1 generation. Then we have meiosis and fertilization. And then you also have in a list your P1 and F1. So again, I'm just going to go through the marks that you guys will get for writing all things down. So you're going to have female, male is when you mention your phenotype, it's 1 mark. Genotype is another mark. Mentioning your gametes, that's 1 other mark. Mentioning the phenotype of the F1 generation, that's another mark. and simply just writing down P1 and F1, as well as Moses and fertilization is another mark. So that is how you get your 6 marks. Okay. Okay. So moving on to the next question, guys. So this question has to do with cloning. So again, with this question is more of just the type of study question. Okay, I just need to again know the content. So, but let's anyway go through it. So it says, a farmer decided to have his best meat-producing bull cloned. The following steps were used in the process. So a muscle cell was taken from the bull, the male, and the nucleus was removed. An ovum was taken from the cow, the female, and the nucleus was removed and discarded. Then the nucleus from the muscle cell was placed in the empty ovum. The ovum was then given an electric shock to stimulate normal cell division to produce an embryo. The embryo was placed in the uterus of a surrogate cow, where it developed into the clone. So this basically just describes the entire process of cloning. Okay, so first and foremost, what is cloning? Simple definition, the production of genetically identical organisms. And then it asks, explain why the nucleus of a muscle cell was used and not the nucleus of a sperm cell. So if we look at the question, what did the farmer want? So this is cloning. So the farmer decided to have his best meat-producing bull cloned. So, okay. So the question says, explain why the nucleus of a muscle cell was used and not the nucleus of a sperm cell. Oh, sorry guys. So what we're going to look at is we are now going to see that the muscle cell basically contains double the amount of the genetic material that we need. So the muscle cell is actually diploid, right? So whereas a sperm cell only has half the genetic material and is haploid. Okay. Then explain why the nucleus of the ovum was removed. Okay, so here we go. So the muscle cell was taken from the bull and the nucleus was removed. An ovum was taken from the cow and the nucleus was removed and discarded. So we only want the best meat producing bull. We only want the genetic information material from the bull. That is why, and we're going to take that genetic material and we're going to put or insert that into the ovum. We don't want any, basically, you don't want any genetic material coming from the cow. Therefore, genetic material of the cow is removed, and we're going to obviously take the genetic material from the best meat producing bull and we're going to insert that into the ovum. Okay. Then state one benefit of cloning. There are a few, but they say one. So if they just say one, just state one because they're only going to mark the first one. They're not going to mark anything else under that. So to state one benefit of cloning, it says you can either produce organisms with desired characteristics or desired traits such as health, appearance, nutritious, yield or shelf life, conservation of threatened species, and to create tissues or organs for transplant. Okay, so that is the cloning part of it. Let's move on to the next part, and that has to do with DNA profiling. Okay, so let's see what it says with DNA profiling. Okay, so a family wanted to identify the biological father of a boy. The mother of the boy is known. Blood groups and DNA profiles of the mother, the boy, and the two males were used to determine paternity. The data below shows the results of the two procedures. Okay. Right. So let's look at what the first question asks. It says, explain why paternity of the boy could not be established using blood groups only. Okay. So the boy. Okay, so let's just change it to a pen quickly. There we go. So it says to explain why the paternity of the boy could not be established using blood groups only. So the mother as is blood group O. So the mother, her genotype is 2 small i's because she has blood group O. The boy has blood group B. Right? So he is obviously blood group B, and his other allele for blood group B would be blood group O, but the recessive allele, because he obviously inherited this recessive allele from the mother. So we know we got his one allele from, but where did he get blood group B from? And that we can't really just determine from looking at the blood groups. Okay. So let's just see how we would write that out. So either male 1 or male 2 could be the father of the boy, since both males have the B allele, B blood group. Male 1 has codominant blood group AB, and male 2 is either homozygous for blood group B or heterozygous for blood group B. So as we noted, the mother's blood group is O. She has the recessive allele and has the homozygous recessive blood group or homozygous recessive allele. The boy would have inherited the recessive allele from the mother, and he would have the genotype that over there. Okay. So who is the biological father of the boy? How do we determine that? So let's just quickly go back to this diagram over here, just to explain it. Okay, so with DNA profiling, you basically are determining the paternity of the child. So in order to determine who is the father of the boy, we're going to look at the bands of both male 1 and male 2, as well as the bands of the mother. So when you are trying to figure out the paternity, the best thing to do is to use a ruler and move down, and you're going to compare the bands by checking the spacing of the bands as well as the thickness of the bands. So first thing is you would compare the boy and the mother. So if you're comparing the boy and the mother, so the boy and the mother, so we can look at the bands. So this band over here, the boy and the mother is the same, as well as that band there and that band there. This band of the boy and the mother is the same, as well as the last two. So there's 4 bands between the boy and the mother that are the same. But now we need to figure out which of the 2 males are, who is the father. So now what you're going to do is whichever bands from the mother and the boy do not align, those are the bands you're now going to compare to see who is the father. So let's start with male 1. So this band over here, different thickness and different spacing. We can't do a comparison between either of these 2 over here. No band to compare over there, no band to compare over there. But here we can compare bands. So between male 1 and the boy, this band's over here. The 2 of them are the same. These 2 bands between male 1 and the boy is the same. And the rest of the bands are not the same, they do not align. Okay, now let's compare male 2 and the boy. So as you can see, this band over here with male 2 and the boy are the same. This band over here and that band is the same, as well as this one and this one and that one and that one. Okay, so we can clearly see that with male 2, there's 1, 2, 3, 4 bands that are the same. With male 1, there's only 2 bands are the same. So because there are more bands that are similar between male 2 and the boy, that will now indicate to us that male 2 is the father of the boy. Okay, who's the biological father of the boy? That's male 2. And I think we also answered the next question, which says explain your answer to question 2.3.2, which asked who is the biological father of the boy. So it says for some bands of the boy's DNA profile match with those of the mother's profile. The remaining bands of the boy match with the bands of male 2's DNA profile, and fewer bands match with male 1's DNA profile. So as you can see, just go back quickly. as we can see over here, there are more bands that match of male 2 that match with the boy, and that's highlighted in the blue over there. And there are less bands from male 1 that match with the boy, as highlighted in red over there. Okay. So let's move on to the next set of questions. So this is state 1, other use of DNA profiling. Again, that's for 1 mark, so you can just state 1. So it says tracing missing persons, identification of genetic orders, establishing family relations, matching tissues for organ transplants, identifying dead persons, criminals, or suspects. So you can name one of those. Okay, moving on to the next section. That is going to be evolution, and that's also one of the bulky parts of the paper, and that accounts for 54 marks. Okay, so what we're going to do is just going to do a bit of a recap on phylogenetic just to explain how they work. Okay. Okay. So we have your phylogenetic tree. So at the start of the year is your ancestor, and as you move towards the end of your phylogenetic tree, you have your present day species. So if we look at A and B, the most recent common ancestor is over here at this point. Also, at this point over here is where speciation events take place, where the line forks where the line basically forks and branches off. That is a speciation event where you can say a new species begins to originate. And then the most common ancestor of A, B, C, D, and E is at this point over here. Okay. So phylogenetic trees don't always look the same. They can either look like this over here, or they can look like this over here. So let's have a look at the phylogenetic tree on the right. So the first branching or speciation event happened at 1. So this species is basically the common ancestor of all the species listed from A to F. So now let's just zoom in on the species at the top. So A, B, and C, they basically all have a common ancestor at 2 over there. And B and C is more closely related to A, and B and C's common ancestor is at 3. Okay. So also where the lines basically end, it is the extinction of that specific species. So, for example, if A was extended all the way to there, as well as C, D, and E, the line of B ended here way before the lines of A, C, D, and E. That means that is where the extinction of species B occurred. Okay. So what's going to happen now with evolution? We're basically just going to cover a few of the important parts of evolution. And we're going to start off with John Baptiste Lamarck and a few of his theories of evolution. So what did he say? So John Baptiste Lamarck's theory wasn't based on 2 things. The first thing or first law that his theory of evolution was based on was the law of use and disuse. So he said that the use or disuse of organs may cause the organs to increase or decrease in size or completely disappear. The next law that he looked at is the law of the inheritance of acquired traits. So basically what he said is that during their lifetime, organisms acquire, that's the important part, during their lifetime, organisms acquire, meaning they gain certain changes in characteristics that are inherited by the offspring. This results in changes in populations and the formation of a new species. Okay, so obviously Lamarck's theories were rejected. Now, why were his theories rejected? First and foremost, there was no scientific evidence to prove that acquired characteristics are inherited. Next, genetics was discovered, and it was found that the gene causes a particular characteristic to occur. So now let's look at the application of Lamarckism. Okay, so the one example, the best known example for Lamarckism is the story of the giraffes. So according to Lamarck, giraffe ancestors had very short necks. And over generations, these short-necked giraffes, they stretch their necks to reach the leaves on top of the very tall trees. And basically, they stretch their necks to grow stronger and longer. And in this acquired trait, the long neck was passed on to the next generation. And as a result, all modern giraffes have long necks. So that was his way of coming about with evolution, or his theories on evolution. But now, obviously, his theory was rejected. But let's look at Charles Darwin, whose theories were a bit more accurate. So, Charles Darwin, his theory of evolution by natural selection. So, what did he say? So, he said there's a great deal of variation amongst the offspring. So, some have favorable characteristics and some do not. So, some giraffes may have short necks, some may have long necks. So, when there's a change in the environmental conditions or if there is competition, then organisms with the characteristics which make them more suited survive, whilst organisms with the unfavorable characteristics which make them less suited, they basically die. So environmental conditions could be change in weather, competition would be animals competing for food. There could be a lot of various factors. So the organisms that survive, that have the favorable characteristic, they reproduce and pass on the allele for the favorable characteristic to their offspring. So the next generation will therefore have a greater proportion of individuals with a favorable characteristic. Okay, so this is basically how you would write out the theory of evolution by natural selection. If asked in the exam, this could also possibly go for about 6 marks in your exam. Sometimes you would have to adapt this, in a sense, generic theory of evolution by natural selection. You would have to then adapt this to the questions that they ask you. The same would apply to Lamarckism. This is also just an example, bit of a genetic example, but also you would have to adapt it to whatever type of question they are giving you. Okay. Okay, so let's move on to a few questions. Okay, so we're now going to look at paper 2, November 2022. Okay. So it says modern day whales are aquatic animals and they spend their entire lives in the ocean. They are thought to have evolved from four-legged ancestors as represented below. So let's look at the first question. So which ancestor of whales most likely lived both in land and in water? So now I just have to look at the diagram and read what is being asked. So they said over here, your Pakicetus, that was a quadrupedal carnivore. Your Ambulocetus, flipper-like large feet, meaning it occurred on land, and Dilson, meaning it occurred in water. You have your Dorudon, large flippers in the front, meaning that maybe they occurred more in the water, and very small hind flippers. So, and then you have your blue whale, non-functioning pelvis, and large flippers at the front, meaning they mostly occurred in the ocean. Okay, so which ancestors of whales most likely lived both in water and in land? That would be the Ambulocetus. Now, the next question asks, give one reason for your answer to question 3.5.1. So they most likely lived both in land and both in water and on land because they had flipper-like large feet for walking and they had a tail for swimming. Okay. So moving on to the next question, they explain why Ambulocetus and the Dorudon may be considered as transitional species in the evolution of whales. So transitional species, just recap that definition again. It says shows intermediate characteristics between two species. So it has characteristics common to its ancestor as well as its descendants. So basically, what they say is they share characteristics or have intermediate characteristics of the ancestor, the Pakicetus, meaning both the Dorodon and Ambulocetus, they both have, so the Dorodon has small legs or small hind limbs, whereas the Ambulocetus has flipper-like large feet, meaning they both can occur on land, right? And they both also have some type of flippers. The one has the flipper, Dorodon has a flipper, and the Ambulocetus has a tail, which allows them to swim. Therefore, they are the transitional species in the evolution of whales. Okay. So then it says, explain according to Lamarck why modern day whales do not have legs. So let's see how we would write it according to Lamarck. So what we can say is that ancestral species of all whales all had legs or lived on land. Okay. Then as more time was spent in the water in search of food, the legs were used less and disappeared. So that's the law of use and disuse. So don't use an organ, it disappears over time. Then the law of acquired characteristics comes in, where the acquired characteristic was passed on to the next generation, meaning that the flippers were now passed on from one generation to the next. Okay, that is now the flippers are now the acquired characteristic. Okay, so that is a bit of Lamarckism and a bit of evolution over there. But now let's move on to the next type. We have to analyze a few diagrams. Okay, so now we're going to look at paper 2, and that is November 2019. So it says the diagram below shows the skulls of two species of primates. Okay, so I have skull 1 and skull 2. So it says 3.1.1, tabulate 3 observable differences between skull 1 and skull 2 that show trends in human evolution. So they said 3 observable differences, meaning what you can see. So you can't write anything that you can't see from these 2 skulls. So it also said tabulate, so you have to draw a table. That's the only way you're going to get your marks. So before we get to the answer, just by looking at it, obviously what I note first is that the skull 1 has a more prognathous jaw as compared to skull 2. Skull 2 also now has a much larger skull or larger cranium. Skull 1 is a much smaller cranium. And then what I also notice is there's more pronounced brow ridges in skull 1 and less pronounced brow ridges in skull 2. So in skull 1, it says the brow ridges, mentions that. It mentions, okay, so it says more protruding jaws or more prognathous jaws, and skull 2 less protruding or less prognathous. And then it mentions smaller cranium size skull 1, larger cranium size skull 2. And then the teeth can be a bit tricky to see, but it does have in skull 1, there is much larger teeth and larger canines, smaller teeth and smaller canines. And then also, you can also take note of the smaller jaws in skull 2 and larger jaws in skull 1. So those for me, I would say, is the more distinguishing or the more prominent observable features if you're looking at this diagram. You could also mention the poorly developed chin or well-developed chin in skull 2. And you also can note that skull 2 is more of a flat face, whereas skull 1 is more of a sloping face, curved face, as you can see over there. Okay, then moving on to the next question. So it says, give 4 characteristics of the upper limbs that humans share with other primates. So this is just a bit of things you have to remember. So when I think of this question, 4 characteristics of the upper limbs, first thing comes to mind is both humans and primates have opposable thumbs. We both have 5 fingers. Both have fingertips instead of claws. Also, both have long arms. So those are the ones that I just think of immediately. The other ones, freely rotating arms, just to mention that, as well as repetition around the elbow joints. Okay. Then next question, it says explain how an increase in cranial volume is related to intelligence. Okay, so obviously we know the cranium has the brain. So obviously if you look at skull 2, you can see it has a much larger cranium size as compared to skull 1. So you can mention that. So basically it also says a large cranial volume indicates a larger brain or more brain cells, which indicates a greater intelligence. So Again, larger brain size or larger cranial volume indicates a larger brain, as compared to a smaller cranial volume indicates a much smaller brain. Okay, so next question, guys. So it says humans are bipedal organisms. What is meant by bipedalism? Just a simple definition, means you're walking on two legs. Okay. Then it says explain how each of the following skeletal structures have contributed to bipedalism in humans. So just a bit of theory you need to know. So with the foramen magnum, that just basically has to do with the spine. So a more forward position of the foramen magnum allows the spinal cord to enter more vertically. If the foramen magnum is in a more backward position, that means the person or the primate might be more quadrupedal. Okay, and then explain how each of the following structures have contributed to bipedalism in humans. The pelvic girdle. So pelvic girdle, to allow for bipedalism, is short and wide to support the upper body, but yeah. Then looking at the spine, how does that contribute to bipedalism? So a spine that is more curved or more S-shaped allows for shock absorption and allows flexible movement and support. If the spine was more C-shaped, that would mean that the primate would be quadrupedal. Okay. Then the last question. This says describe the process of speciation through geographical isolation. So this question accounts for 6 marks. So with this question again, you also need to mention it in chronological order in order to get your marks. And again, if you don't, then you won't get the full marks. And also, this is a very, very generic way of mentioning geographical isolation. Obviously, sometimes you have to specify the process of speciation through geographical isolation. Sometimes you have to specify that to the question provided. Okay, but let's just go through the process of speciation through geographical isolation. So it reads, a population of particular species becomes separated by a geographical barrier. So species A, species B. So, okay, so a population of a particular species becomes separated by a geographical barrier. So that happens that there is no gene flow between the two separate populations. Okay. So natural selection occurs independently in each population due to exposure to different environmental conditions or selection pressures. Then the populations become very different from each other genotypically and phenotypically. And when this happens, even if the population were to mix again, they will not be able to interbreed because the different populations are now known as new species. Okay. So guys, that is actually the last slide of this presentation. So I would actually like to thank you guys for joining us for our final lesson of our spring school. I hope you guys learned a lot through all the sessions throughout this week during the maths, physics, and life science sessions. And we would like to wish you guys all the best on your upcoming exams. And we'll see you guys again in end of November for that final prep before your final papers. So thank you guys and all the best for your studying. Hope you guys enjoy the session and yeah, enjoy the rest of your holiday, guys.